Hi, this is a quick demo of DNA Barcode Browser, which is an entry in the GBIF 2020 challenge. So what I'm interested in is ways to explore biodiversity data, particularly DNA barcode data, using the fact that we have these sequences, and sequences mean that we can do some interesting things that we can't necessarily do with traditional kinds of biodiversity. So we have more information here than just a taxon name, a location, and a date. So what you can see in the web page for this app is on the left-hand side, you've got a little widget with a DNA barcode. You have a map which displays distribution of DNA barcodes. So underlying this this website, there's a database I've created from DNA barcodes, a sort of DNA barcode data which is in GBIF. In this case, I've taken it directly from Bold, the Barcode of Life, rather than go via GBIF. So let's get started. So one thing we might be interested in is, say I've got a DNA barcode, like this one here. Where does that barcode come from, and what other barcodes look a bit like that? So I'm just going to go and click on the search button here. So we're going to go and search for the barcode in this database, and we come back with a set of barcodes, and we build an evolutionary tree. And this tree is built on the fly in your web browser. So each one of these things here is a DNA barcode, and there's a couple of things to notice about this tree. It has a very characteristic shape. The center of the tree is basically empty. The barcodes are all kind of scattered around the rim, and many of the edges in this tree are very short. What this means is that most of these barcode samples come from the same species, or in this case just a few different species, and they're all very closely related to each other. And where do these barcodes come from? Well, they come from this part of New Guinea here, and these barcodes are for a group of fish, and we can see that from the sort of classification we've got on the left. So this is the taxonomic classification of those barcodes, and there are some pictures here which you might just be able to make out pictures of fish. These are rainbow fish. And you can see here are the species names. Each one of these blue links is the actual barcode sample ID, and it's a link back to the Barcode of Life database. Now, just going quickly back to the tree. So we have the evolutionary tree. You can sort of have a play. You just zoom in and out with your mouse and have a look. You can download the original sequences. They're not aligned. The way this technique works is it doesn't need sequence alignment. It just grabs the sequences and builds a tree. The distances between the sequences, you can download those, and you can download the tree as well, and have a look at those and some software if you want to. There's a little number here called the tree length, which for this tree is 0.092. And the tree length is basically, we just add up all the branches, all the lengths of these branches all around the tree. And it gives us a measure of how much diversity there is in that tree. So one approach is we have a sequence and we search this data using that DNA sequence. Let's imagine we wanted to search geographically. Well, we can do that by having a look at this map, and let's say, okay, let's go and grab there. What barcodes are there? What are they for? Now, it turns out that these barcodes are for bats. There's a group of barcodes from bats in Vietnam, and here they are here. You can see them in a bit more kind of detail. Now there's our tree, and again, it's one of these trees with this very characteristic structure that you often see with barcode data. Yawning gap in the middle. So these are lots of barcodes from, in this case, these are all from one species here. The overall diversity of barcodes is fairly low. The tree length is 1.92. Now you could ask, well, okay, let's go to maybe a slightly different part of the world and see what's there. So I'm going to go and have a look at this region here, and I'm going to draw a box that's roughly the same size. And let's see what's there. Okay, now this is very different. So again, we've gone and talked to the database, we've grabbed some barcodes, and we've built a tree on the fly. But look at this tree, it's very, very different. So the length of this tree is much greater, it's 7.5. And the center of the tree is no longer empty. This is the kind of pattern you get when you've got barcode samples from lots of different species in the same area. So some of these species are represented maybe by a single barcode or just one or two. sort of clustered around the edges of the tree. But in the middle here, this tells you that these barcodes are from different species, different genera, maybe quite distantly related. So there's clearly different patterns in these two samples. And you could start to think about, well, okay, maybe we could use this as a measure of biodiversity using this phylogenetic structure. So we've got two different regions in Southeast Asia. We've sampled roughly the same size area. We've got very different results. Now, obviously, in this example, there's probably all sorts of reasons why we have different samples. But there's a lot of potential here. You could imagine, dividing the world up into a grid of roughly equal sizes, sampling the barcodes there, measuring phylogenetic diversity, what kind of tree structure do we have, and using that as a way to assess biodiversity. So that is this app in a nutshell. It's the idea of trying to explore biodiversity using barcode data in a way that's kind of native to that data. So either you can do it geographically and have a look, or if you prefer, you can explore using sequences. So we can search this kind of data using sequences and build a tree. So I guess what I'm hoping is that this kind of approach looks appealing, and you could imagine that in future you could go to GBIF and search and explore the data not only geographically but also using sequences. Thanks very much for your time.